Yes. Now we will be talking about the acquired immunity. Acquired means we have acquired it. We have taken it from the our exposure to the various antigen. Now antigen have lots of types. So out of that, whatever immunity we have adapted, that is called as the acquired immunity. It is very much specialized than the other immunity mechanism. What is the other immunity mechanism? That is the innate immune response. So it is very much specialized. Means innate immune response we have seen has a special general mechanism. They do not means they do not have any type of memory. They just kill. So when we talk about the acquired immunity, it is simply a resistance that individual acquires by exposure to various pathogens or the microorganism during the course of life. What it means? That means we will have a specialized resistance. Resistance means that particular time through through the exposure, our immunity builds the memory against that particular organism, and throughout our life, we will not get diseased by that particular organism. That is called as the resistance. Now, when we talk about the acquired immunity mechanism of action, so it has two specialized mechanism of action. The first is cell mediated, or second is the humoral. The term humoral means something which is soluble into the blood. So these two means of acquired immunity interact and destroy whatever foreign antigen has invaded in our body. Alright. So when we talk about the characteristic, we know the first and the most important characteristic which differs the acquired immune system from the innate is the specificity. We have seen that innate immune response is nothing but the killing the microorganism in a generalized way, but acquired immunity is not like that. It has the specificity. That means it will kill the microorganism when it is properly processed and it is specific, or body memory cells, that means acquired immune system players know that particular microorganism. Understand? So after the recognition only it will act on that. It is not like that like innate immune mechanism, it will work in a generalized way. So it is highly specific. When we talk about the B cells, so you will find that more than lakh identical antibodies one B cell can produce. So that means if your body is exposed to the lakhs of microorganism or lakhs of pathogen, so all the pathogen differ from their antigenic property, right? So this way they are going to be treated by the various antibodies by the B cell. Now when we talk about the immune diversity, diversity means having the various forms. So when we talk about the immune diversity, so if it is specific, so special antibody will be formed for the special type of the antigen. So tremendous diversity of antibodies will be found. It is believed that more than 10 as to 8 different B cells are present into the bone marrow, and they are responsible, and one B cell we can see approximate lakhs of antibody can be produced. It is having the immune memory. It is not like that they are processing the microorganism, and if the microorganism will have the re-exposure again they will start the same mechanism. It is absolutely not like that. They build the antibody. That antibody design is saved forever, means they make the memory cell. So this is called as the immune memory. For example, if a person is infected with the malarial parasite, so that malarial parasite will be processed, and the antibody against the malarial parasite will form. Not only this, but the memory cell which can recognize the malarial parasite re-exposure will also be formed, and it will be saved for the lifetime. So if the same person will have the exposure to the malarial parasite next time, will not be getting any type of disease, because it will adapt a resistance against the malaria. So this memory cell will capture that malarial parasite or will kill them. Is that clear? And this is so accurate that most of the time our body will not have a single symptom of entry of that particular parasite. Understand? The best part about the adaptive immune response is that it can recognize self and non-self. This is the best part. It means they have exposure to all the bodily cells while their development. So they means the primary immune organ. They checks that whatever adaptive immune system cells, that is the lymphocytes, are formed, they are capable of recognition of self and non-self or not. When we talk about the acquired immune response, how it is initiated, so it is because see, generally we all have talked till now about the innate immune response, and in innate immune response, what we have learned that generally if anything is entering in the body is handled by the innate immune response in a generalized mechanism. So how body will switch to the acquired immune system? So it will switches by recognition of antigen by lymphocytes checks whether this antigen is new or it is having the re-exposure. Then when if it finds that this is something new, so this will activates the lymphocyte, but directly this antigen will not interact. Directly, the antigen cannot talk or cannot be recognized by the lymphocytes. For that, they require some type of the helper molecule. These helper molecules are known as the major histocompatibility, that is MHC complexes. And once the activation of this lymphocyte is done, after activation they differentiate means they form, as I told you, a single B cell will be having the lacks of antibody on it. So out of that, only a single receptor which is required for that particular antigen to kill, or the specific designed antibody which is required to kill that particular antigen will be formed. And once the perfectly suited B cell is ready, that will proliferate. Proliferate means it will increase in number, it will multiply and will turn into the effector cell. Effector cell means the cell which is going to act. Now this effector cells will eliminate the antigen completely. See this, this is a generalized special mechanism which is helping the effector cell to eliminate the antigen. But sometimes, if especially when the T lymphocyte or C type of lymphocyte, cytotoxic C cells, which is also called as the T killer cell, is involved. So sometimes they kill the self cell. Now you will be thinking, ma'am, they are well versed to know what is the self and non-self. Then how do they kill? They kill the infected cell, the cells which are either infected by the virus or the bacterial infections, or the cell has transformed and trying to be convert into the cancer cell. Understood? So overall, for the removal of the antigen, they eliminate some body cells also. That's why they are getting sometime swelling, edema kind of condition when the immune system is active. Not only this, after doing all this operation, they save the memory cells कि ये पूरा मेहनत दोबारा ना करना पड़े. This memory cells elicits a rapid and long-term response. I told you this memory cell will remain in the body forever till your last breath, and whenever this parasite will respond or re-expose, they will kill that and they will eliminate that. Most of the time, without knowing your body, you will not have any type of infection. Now, as I told you, this immune system, that means your adaptive immune system, have two ways of mechanism. We will talk about the first, that is called as the antibody mediated or the humoral immunity. Why antibody mediated mechanism is called as humoral immunity? Because this antibodies, which is also called as the immunoglobulin, is dissolved or it is present into the soluble form in your blood. I had already told you the term humoral means mix or soluble into the fluid. So this antibody, which are the proteins, immunoglobulin, which is soluble in your blood, that's why this particular type of response is called as the humoral antibodies. Now, which type of cells are producing the antibody? Lymphocytes. We have two lymphocytes. One is the T lymphocyte, another is the B lymphocyte. So these antibodies are produced by the B cells. Now, how to get they activated? How the B cells are getting information that I do need to prepare the antibodies? So directly, as I told you, that lymphocytes are very specific. They do not recognize the antigen until and unless they do not have any information about it. So they are getting stimulated by the antigen, that is correct. But for that, they required the official antigen presenting cells. These cells are also called as APC. APC means antigen presenting cells. So when any antigen is processed properly and brought by the antigen presenting cell, so this will be talking not directly with B cells. This will interact with the this MHC1 molecule will interact with the T helper cell. T helper cells. Now, how do they talk? I told you they have the specialized molecule that is called as interleukins. So they will interact with the T helper cells with the interleukin 1. And this interleukin 1 will tell the TH cells that this is the process antigen, and then thereafter this T helper cell will take a decision that I must go to the TC cells, that is the T cytotoxic cells, that particular things to be killed by the toxic effect, or it requires the antibody. When the T helper cells takes the decision that antibody to be produced, so it will go to the B cells and will tell B cell to produce the new designed antibody for this particular antigen. Now, how do this antigen and antibody recognize each other? So the particular region which is binds to the antibody and antigen, which is called as the paratop, that is also known as the FAP segment. FAP segment antibody का एक special region होता है that will bind to the particular antigen, which is also called as the paratop, and the region which is crystallizable, that is called as the FC region, which is responsible for the effector function. Same way, antigen also have the determined region which is antigenic in nature, that is called as the epitope, and this epitope will be checked by the antibody. Epitopes are the specialized region which is responsible for stimulating the antigenic response. Okay, I told you what do you mean by the antigenic response. Antigenic response means eliciting or challenging the immune system. So that particular region will be recognized by the B cell, and B cell will form. It is having the mutated region which can form a specialized fit molecule that is called as the antibody. So this antibody will lock and form the this antibody can kill that particular microorganism in a various way, and it will work as first thing agglutination. See, students get always confused between the term agglutination, then precipitation, and the coagulation. These three terms have the meanings formation of the complexes, but with a little difference. Term agglutination is very important for your immunology topic. Agglutination means forming the solubilized complex. Solubilized complex means these are present in a colloidal form of your blood only. Is that clear? Precipitation, a complexes which is formed by the chemical reaction and having the property to settle down. Understood? And the coagulation simply forming the clots. So here, what is the property of the antibody? They can form the agglutination. They have the property which causes the cells to clump together, and this antigen coated with the antibody cluster together. Now which cells? The antigenic cell. They will cluster together. They will stick to each other because of the presence of the antibody over it, and which will make them immobilized and vulnerable to phagocytes. It's a kind of locking them. Second term, or the second property is opsonization. What do you mean by opsonization? This antibody will bind to the antigen and will lock them, will not let them to, it is some kind of the imprisonment. Imprisonment means they are locking them, they will not letting them go anywhere. And what will elicit by it? Phagocyte will get signal, yes, something is trapped here that should be removed, and then phagocytes will come and kill that particular antigen, which is known as the opsonization. So generally, what happens in the case of the opsonization? Coating of the bacteria by the antibodies fabrication. Neutralization, we know there are certain type of the bacteria which can release the toxins. So these toxins can give the harmful effect to our body. Antibody, what they do, they cover the toxic site of that particular antigen. Example, if we will talk about the tetanus toxin, that will be neutralized when IgG antibody binds. So that toxins will be covered by the antibodies, so they will neutralize them. The fourth property of antibody is complement activation. Complement activation means punching the hole into the cell wall of the bacteria. Especially this particular type of immune response is available for treating or handling the bacteria. So what does it do? It simply punches the hole in the cell wall. So when the hole is drilled into the bacterial body, what happens? Osmotic imbalance occurs. Osmotic imbalance occurs means it leaks the bacterial bodily content, and because of the osmotic imbalance, the bacteria simply dies. Antibody dependent cell mediated cytotoxicity. Antibody mediated cell dependent cytotoxicity means antibody binds to the organism via their Fab region, and this large granular lymphocyte natural killer cell attached with the FC region and kills all this organism by phagocytosis, and it released the toxic substance which is called as the perforin. So this is called as antibody mediated cytotoxicity. It is called as because antibody is involved for cytotoxicity of that particular system. Now we will be talking about the next mechanism of action, that is called as the cell mediated immunity or T cell mediated immunity. Cell mediated name itself suggests that CMI will be having cells as a player. In the humoral immunity, the main player was antibodies. Cell-mediated immune response caused by the T lymphocytes. T lymphocytes. Now, B lymphocytes role we understood. The most important role of the B lymphocyte is the formation of the antibody. Same way, T lymphocyte can also having the specific action. That means they will recognize the foreign invader by surface receptor, or they also have the TCR, T cell surface receptor, but when the microorganism is processed by the MHC molecule, then they can talk with the lymphocyte. Now T lymphocytes, they can attack and destroy invaders directly or by releasing the soluble cytokines. Now how can they do? What are the principal kind of the T cells? T cells, we have various types when we talk about. So first type is the inducer T cells. Now, inducer, that means they are going to induce other cells to activate the immune system, and this immune cells will start dividing. Then second category, helper T cells. These are the most important type of the T cells. This will initiate the humoral immune response by stimulating B cells to produce antibody. Just now we had discussed about them. Then cytotoxic T cells. Cytotoxic T cell is also called as a killer T cells. These are the most important cells which have important role in CMI, that is cell mediated immunity. Now this kills the host cell which are infected with the viruses or bacteria. It can also recognize and kills the cancer cell. The next category is the suppressor T cell. Suppressor T cell, what it does, it terminates the immune response. Now you might be thinking, okay, ma'am, why do we need to terminate the immune response? That means when the body immune system is the very smartest system of our body, when the body recognizes and confirms that the antigen is completely eliminated. So these killer T cells should not harm the fresh cells or non-infected cells. So this PS cells or the suppressor T cells stops the TC cells. Now don't function, now don't work, stop now. So this immune system will be terminated. T helper cells particularly most important because they maximize the capability of the immune system. Now when we talk about the T helper cells, they cannot destroy infected cells, they cannot destroy the pathogens, but they can activate and direct other immune cells to do so. That's why they are called as the helper T cells. Means they will pick up the antigen and they will check whether the antigen is properly processed by the APC, that is the antigen presenting cells. We had already discussed that APC involves the macrophages, dendritic cells, and all the nucleated cells. So this APC will whether they have processed the proper antigen or not. This checking part is done by the T helper cells. T helper cells can relate that antigen or can inform the other type of lymphocytes, that is the B lymphocyte as well as the T cytotoxic cells, that the antigen has come. Now you start working on it. So the major role of the T helper cells to stimulate B cells to secrete antibody and to activate the phagocytes, to activate the T killer cells and enhance the activity of the natural killer cells. So, what is the major function of the T killer cells? T killer cells activate the destruction. Now, T killer cells recognize and destroy the infected cells. Now, if the cell is infected by virus, if it is transformed, so all this can be killed by the T killer cells. They can also play an important role against the intracellular bacteria and certain types of the cancer. Now, you might be thinking, ma'am, natural killer cell is also doing the same. Yes, natural killer cell will be doing it into the generalized mechanism. But T killer cells, if the particular virus is processed properly by the APC, so that time the T killer cells or the T cytotoxic cells will play a role. Now T cells have the antigen receptor for the recognition and react to specific antigen. It is not like that, okay, every time they need to process. They will also have like B cells, the receptors. So if the same virus or the same bacteria will enter in the body, they will elicit the immune response directly. Understand? So for that, they should have the prior knowledge. T cell receptor only recognize antigen combined with the MHC protein on the surface of the cell. After the macrophages has processed the antigen, it releases the interleukin 1. We had already discussed that the MHC molecule and the T helper cells, they talk with each other with the interleukin 1, and T helper cells talks with the B cell or other lymphocyte, that is the TC cells with the interleukin 2, which stimulates the multiplication of the cytotoxic cells. Now body cells have been infected by the antigen that are destroyed by the cytotoxic T cells as the infection subsides. Now, suppressor T cells turn off the immune response. So, this is all about the mechanism of action of the B cell and the T cell. At this note, we'll stop here.